Good morning, and thank you for joining. I am Katie Bockrath, a geneticist with the U.S. Fish and Wildlife Service, and today I'll be presenting the results of a pilot study investigating the microbial community of the freshwater mussel. This work is a collaboration between the U.S. Fish and Wildlife Service, Midwest Fisheries Center, and the USGS Upper Midwest Environmental Science Center in La Crosse, Wisconsin. What is a microbiome? A microbiome is the community of microorganisms living together in a particular habitat. Humans, animals, and plants have their own unique microbiomes, but so do soil, oceans, and even buildings. The microbiome is a symbiotic relationship with the host, where each benefits from each other. In humans, the microbiome plays a role in many different biological functions, from breaking down food and synthesizing vitamins to aiding in tissue growth and pathogen resistance. The microbiome is a critical component of our bodies. Microbiomes are complex, ever fluctuating and unique. The diversity and relative densities of microbial species differs, not only between individuals, but across tissues or biological compartments. It changes as we age, with the changes in the environment, with our diet, and with the medications that we take. The biological functions the microbiome provides and the variation in its makeup is not only seen in mammals, like mice and humans, but in bivalves too. Shown here is the microbial community of the black-lipped pearl oyster across multiple biological compartments. The figure on the left, the y-axis, shows the percent composition of the bacterial community of each biological compartment. Different bacterial species are represented by different colors. We can see there's a lot of bacterial diversity within each biological compartment, but more importantly, that the biodiversity varies across each compartment. The microbiome of the gill is distinct from the microbiome of other biological compartments. This is reflected in the figure on the right, which shows a principal component analysis of the same bacterial diversity of each biological compartment. Additionally, when the bacterial species themselves are identified, similar to what has been observed in human microbiomes, the oyster microbiome plays a role in daily biological functions and immune response. For instance, microbes associated with nutrient acquisition and antimicrobial activity are found in association with the gills, which are highly exposed in the environment and involved in filtering out food. So, if microbiomes are important for humans and marine bivalve health, what about freshwater mussels? Do the microbial associations we observe in oysters hold true in freshwater mussels? Not much is known about freshwater mussel microbiomes, but from other studies, the microbiome should play a large role in overall mussel health in response to changes in the environment. In this study, we aim to identify what the baseline microbiome looks like for 4 mussel species across multiple biological compartments. We sampled from a long-term studied mussel bed in the upper Mississippi River that has well-documented mussel species composition, recruitment, and survivorship. So the microbiome can differ in many ways, and we had a lot of questions about how the microbiome might differ between things like species, tissue types, habitat, seasons, life stages, and health status. For this pilot study, we focused on species, biological compartments, and habitat. Our sampling location was West Newton Chute in Pool 5 of the Upper Mississippi River. This location is surveyed yearly and has had 26 observed species across the 10+ years it has been surveyed. Some species are rare and not observed every year, while others are more abundant and are observed in large numbers every year. For this study, we sampled from 4 abundant mussel species, the pimpleback, the wabash pigtoe, three-ridge, and the three-horned wartyback. West Newton Chute bed is broken up into 4 sub-areas. The A and B core areas are characterized by high mussel density at 11 mussels per meter squared and high survival rates, while C and D periphery areas are characterized by low mussel density at 0.5 mussels per meter squared and lower survival rates. Each of the 4 mussel species was sampled from all 4 sub-areas. For this pilot study presented here, we focus on core area A. Previous studies have shown that microbiomes differ between biological compartments. Because the microhabitats for bacteria on the gills is probably much different than the microhabitat of the foot, it stands to reason that the microbiome of each would differ. To get a baseline of the mussel's overall microbiome, we sampled 4 biological compartments: the gill, the foot, excurrent siphon, and hemolymph. The environment has its own microbiome, and it should differ from that of the mussels and each of its biological compartments. 3 environmental samples were taken in relation to how the mussel might interact with the environment. At each sampling location, we took sediment samples, water from just above the sediment, and extracted the pore water from the sediment samples themselves. DNA from all the environmental samples was extracted using phenol chloroform, and the biological samples from the mussels was extracted using an isohelix extreme DNA extraction kit. 2 standard microbial genetic markers were used to sequence the microbiome. Bacteria were targeted using 16S, and fungi was targeted using ITS. All samples were PCR amplified for both genetic markers and sequenced in 2 directions at 300 base pairs. Sequences were analyzed using QIIME2 microbiome pipeline. The figure on the right shows a rarefaction curve for all samples sequenced. The x-axis shows the sequencing depth, or the number of sequences that were collected for each sample. The y-axis shows the observed OTUs, or unique sequences. Given the biodiversity in each sample, sequencing depth was sufficient, indicated by the curve's plateau occurring well below the maximum sequencing depth for each sample. Given the extensive sequencing depth, there should be enough sequencing data to capture rare or low abundance species. Overall, there's a significant difference between the mussel microbiome and the environment and the microbiome of the different biological compartments. In the table to the left, we see the beta diversity of the microbiomes between the 3-ridge and the 3-horned wartyback, and the beta diversity between each mussel and the environment. There's a significant difference in the microbial diversity between the mussels and the environment, but there's no significant difference between the mussel species themselves. The table to the right shows the differences in beta diversity between each biological compartment. All non-significant differences are highlighted in gray. The diversity of the environmental samples were not significantly different from each other, while for the most part, the diversity of the biological compartments were significantly different. This figure shows the combined bacterial microbial diversity for both 3-ridge and 3-horned wartyback and the environmental samples. The figure is broken up by biological compartments with relative frequency on the y-axis. Each vertical is an individual sample, and the different microbial taxonomic IDs are shown in different colors. The microbial taxonomy is to the class level. There's a lot of biodiversity present within the biological compartments and within each sample, but the biodiversity between biological compartments is different. If we break this down and look at individual trends in taxonomic groups, we see that blue-green algae are associated with the gill and the excurrent siphon more than any other biological compartment. Additionally, there's a group of bacterioidea that are only associated with the foot samples, while there are other groups that are only associated with the, there are other groups that seem to be ubiquitous across all the samples. By looking at the species level, the biodiversity increases, and we get a bit more resolution on some taxonomic IDs. For instance, some of the blue-green algae associated with the gill and excurrent siphon are actually freshwater diatoms. So overall, some taxonomic diversity takeaways. The microbiome is different from the environment. Differences in taxonomic diversity are seen across biological compartments. The taxonomic groups may be unique to a biological compartment or only found in association with mussels. For example, algae and diatoms predominantly are found in association with gills and excurrent siphon, but not with any of the other tissue types. These are all consistent to what has been observed in marine bivalves. So like I said earlier, this is a pilot study to a greater effort. In the future, we're going to expand on this pilot study by including all 4 species that were sampled at West Newton Chute across all of the biological compartments, including both the core and the periphery samples. Additionally, we're going to incorporate the fungal ITS sequences. Also, we'd like to do some additional field sampling. The periphery samples were low abundance, and we did not meet our target for all of our species in the periphery. Additionally, we'd like to use this baseline microbiome to compare to other studies. Some additional studies that we'd like to compare to might be studies looking at disease state versus non-disease state, how hatchery microbiomes differ from wild microbiomes, and how relocation and introduction efforts could be affected by differences in microbial communities. And with that, I'd like to give a big thanks to Teresa Newton at USGS Upper Midwest Environmental Science Center, Maren Tadalau with the U.S. Fish and Wildlife Service Midwest Fisheries Center, to Bernard, Zeb, and Mike, and the whole mussel dive team with the Minnesota Department of Natural Resources. Without everyone's effort, especially the dive team, this project would not be possible. With that, thank you.